All right, thanks everyone for sticking it out to the end. There's nothing like ending a conference with a really cheerful topic like impacts of drought on fish populations, but probably should have ended with David's talk. Anyway, I'd like to just acknowledge my co-author on this, Andrew Bartshire, who's responsible for all the maps that you're going to see in this presentation. In keeping with Cleo's theme of collaborative science, I'll give a little background on our program. It's been a collaborative effort from the beginning. We started working with Department of Fish and Wildlife, the Army Corps, National Marine Fisheries Service, and the Water Agency, Sonoma County Water Agency, to develop and implement a coho monitoring program to help inform the Russian River coho salmon captive broodstock program. So our monitoring began in 2004 surrounding evaluating that program. More recently, we've been partnering with the Sonoma County Water Agency and Department of Fish and Wildlife on implementing the California Coastal Salmonid Monitoring Program in the Russian River. And we've also been involved with the Russian River Coho Water Resources Partnership, which is a group of nonprofits and resource conservation districts and different agencies to work on specifically on this issue of flow and the impacts of low flow on endangered coho populations. And that program is funded by National Fish and Wildlife Foundation. We're located in the lower part of the Russian River watershed. The streams that are highlighted in blue are considered potential coho salmon habitat. This is our sampling frame for the Coastal Monitoring Program, or CMP, statewide monitoring program. And so we use a subsampling approach, the GRITS sampling design, to look at status and trends basin-wide. But then we also do some more intensive monitoring in these watersheds that are highlighted. The Dry Creek watershed is our coastal monitoring program, life cycle stream, and then these other 4 that are highlighted in gray are part of the coho broodstock program monitoring. The data that I'm going to show today is focused on these reaches that are highlighted in this orange color, and it's not a random sample of all of the coho habitat in the Russian. These are reaches where we've been looking specifically at flow, and we've been doing a lot of fish monitoring. So I'll be focusing on those because that's where we have the data for the last 3 years. And I'll be kind of narrowing in on and giving some examples of this watershed that's highlighted, Mill Creek. Just to give you some background on the kind of data that we're collecting, during the winter season, we do red surveys between November and mid-April. So we follow the statewide CMP protocols for that. We walk the streams on a 2-week cycle, and we're basically counting and identifying reds to species. And we collect spatial data as well, so we can look at the distribution of reds according to species. And this is just an example of Mill Creek of the kind of data that we're collecting. Then during the summer season, we go out and we do snorkeling surveys as a follow-up to see whether spawning was successful. Again, we're following statewide protocols. We dive every other pool and count and identify juvenile salmonids to species. During the last couple years, this is what a lot of our surveys have been like, very hot and dry. And again, we're collecting spatial data as well. So this is just an example showing the densities of fish. So the larger the size of the dot, the higher the density of fish. So in this case, mill flows from left to right, and so you can see higher concentrations in the lower part of the watershed. And then in the interest of trying to compare our this data with what's going on with flow, a lot of times as fish biologists, we try to compare all these fish metrics to what's going on at one gauge in a stream or a couple of gauges. And in these, coastal California streams, flow gets so low in the summer, it's often really, really hard to measure. We're talking sometimes 1/10 of a CFS. And so that flow gauge data is sometimes not enough to really get a handle on what the fish are experiencing. And so we've really started to turn towards these low flow surveys to generate wet-dry maps. Derek talked about this. It's really simple. You go out with a GPS unit, you walk the stream, you record where it's wet, where it's dry, and where it's intermittent. And it generates these maps that you can compare from year to year and stream to stream. So red is dry, blue is wet, and the yellow is intermittent. So this is 2013, 2014, a little more red in there, 2015 kind of similar. So by going out doing these surveys every year across streams, we can kind of get a better handle on what the fish are experiencing. So this figure is kind of summing up the data that we've been collecting for the last 3 years and looking at it in terms of proportions. So each bar represents the proportion of the stream that we surveyed that was dry, shown in red, blue, shown in wet, wet shown in blue, and then the dotted is intermittent. And so, the first thing that just kind of jumps out at you is there is a lot of red in this graph. So a high proportion of all of these streams in the last 3 years is going dry. And another thing just to notice is there's some differences among the streams. A stream like Dutchbill with progressive drought has remained fairly consistent in terms of the habitat that remains wet, whereas a stream like Pena Creek, we see progressive drying across years. And then there's just some streams that you just don't want to be in if you're an aquatic species during drought years. So one of the questions we've had is, okay, this adds a level of quantification to the amount of habitat, wetted habitat that's available to the fish. Are they able to find these refuges? Are fish spawning in those parts of the stream that remain wet? Are the juveniles rearing in those parts of the streams? Or is their distribution kind of, similar to what's actually available? And so to do that, we've been bringing these different spatial layers together to be able to evaluate it. So we're bringing, in this map, we're bringing together our red survey data with the wet-dry mapping. And so what you're seeing here is the triangles are the reds that we observed in 2013. And again, this is upstream. This is the mouth of Mill Creek. So you can see the distribution. The colors represent the habitat condition the following summer. So we're really looking at, are these fish spawning in places where their offspring are going to experience wetted habitat conditions without having to move around? And what we've found in this year is that 57% of the reds that we observed were in locations that were dry the following summer. And it was even higher percentage the following year. Almost 70% of the reds were in places that were dry. And so unless the juveniles were able to move into places that remained wet, those fish had a very low chance of surviving. And so this is kind of the same idea, but it's comparing with our snorkeling surveys. So we do our surveys between June and August, and that's really a time when fish stop moving around. We've done a lot of tag antenna work, and in the summer there's very little movement. So these fish are kind of stuck where they are when we do our snorkeling surveys. And so what we're seeing here is fish, the coloring is representative of what the fish experienced at the end of the summer. So the higher densities are shown by the size of the dot. So again, just really high concentrations of fish rearing in areas that went dry. 77% of the juveniles we counted in July really had no chance of surviving the summer. And pretty similar results the following year, almost 70%. So just to kind of sum all that up and look at it in terms of proportions, starting with the bar on the left, that's showing what's available. We surveyed in Mill Creek, we surveyed about 18 kilometers of stream, and about 60% of that was dry. And, what we're really seeing is that, the majority of the fish are not making it to these refuge areas. A lot of them are spawning in these areas that go dry, and the juveniles are not able to make it to these refuges where it remains perennial. And I've been focusing on Mill Creek, but summarizing all of the streams, we looked at 5 different watersheds, 45 kilometers of habitat, and pretty similar results. These fish are just not rearing in locations where they're going to survive in these drought years. So I don't really want to end with complete gloom and doom. There are, I think, with the drought, there's been a lot of awareness about how much of a problem flow is for these salmon populations. And one of the groups that we've been involved with is the Coho Partnership. This is a group of nonprofits and RCDs working together, taking a science approach to looking at where flow is an issue, which areas are important for fish, and then working with landowners to try to find alternative ways of getting the water they need by storing water that's highly available in the winter so that they can use it in the summer and not divert from the streams. So that's been a great program, and there's been a lot of other really good work. David talked about the VDI program, so a lot has come out of that program, and there's been some emergency measures too. A lot of the fish that were in the red zone of those maps that I shown actually were relocated. Department of Fish and Wildlife had a kind of massive fish rescue operation this year, and a lot of those fish were actually relocated to the hatchery or to other perennial stream locations. But to me, those are Band-Aids, and the longer term solutions are going to be these alternative ways of storing water. And I think it's great that there's a lot of attention on that right now, and I hope that we see that continuing beyond just these extreme drought years, because that's what it's going to take. I think this flow issue is such a big deal that if we don't really take it on, we're not going to see self-sustaining populations of fish in this watershed. So again, very collaborative program, many agencies and landowners and organizations involved in this work. Thank you.